My name is Charles Farnsworth, and I'm a scientist at the PTMScan group at Cell Signaling Technology. The data I'd like to present today involves two enrichment methods for phosphopeptides. The first is the use of phosphospecific antibodies, alone or in combination, and with the metal affinity chromatography called IMAC. The method here, in Figure 1, is we'll be using gastrocarcinoma cells treated with a control DMSO, a MET tyrosine kinase inhibitor, SU11274, or staurosporin, which is an inhibitor of the PKC family of protein kinases. Following treatment of the cells with the inhibitor, the proteins were purified, digested with proteolytic enzymes, and the purified peptides were then enriched using phosphospecific antibodies or IMAC. In Figure 2, we can see that there's a list of the phosphospecific antibodies that were used in this study. At the top of the list are the antibodies in the basophilic category. As you can see, they require arginines and lysines nearby the site of phosphorylation. We also have a group of proline-specific antibodies. They require proline near the site of phosphorylation. And then all these antibodies together make up a serine-threonine mix. In the middle here is the phosphotyrosine antibody. We use that alone. The method here for PhosphoScan involves the isolation of the peptides, purification of the peptides, and then central to the method is the isolation of the peptides using antibodies linked to agarose speeds. For the IMAC portion of the enrichment method, the same peptides will be used challenged against the metal affinity column here. Following isolation and purification of the phosphopeptides, they'll be run on LCMS, and the data will be processed for both qualitative and quantitative information. It's important to know here that we're doing quantitative proteomics, and we'll be identifying both the peptide and the parent protein. Now, one of the important aspects of using antibodies is you'll be able to pre-screen your samples. So for example, as we can see in Figure 4, we have Western blots for in the first panel and panel A using our phosphotyrosine-specific antibody. And here we can see, as compared to control, lane 2 shows a significant reduction in phosphorylation, which is consistent with the inhibiting of MET. In lane 3, we can see a more modest reduction in phosphorylation, and that's to be expected because staurosporin is not a receptor tyrosine kinase inhibitor. If we look specifically at phosphorylation on the receptor tyrosine kinase MET, we can see in panel B that phosphorylation of MET is almost completely reduced, while staurosporin has not affected MET phosphorylation. And then in panel C, we have a multiplex Western blot using 5 phosphospecific antibodies and a loading control. And we see some interesting features of using 2 different inhibitors. We can see that AKT in lane 2 is significantly reduced in phosphorylation. And also the MAP kinase ERK. And we can see in lane 3 that there's actually a modest increase in phosphorylation and ERK signaling, suggesting a known crosstalk between the 2 signaling pathways. It should also be noted that phospho-S6 is reduced in both experiments as to be expected. Now, the phosphospecific antibodies that we're using can also be used for Western blotting. And in the next figure, I'll show you some specific results using the same antibodies we used in peptide capture. So for example, in Figure 5, we can see in panel A, the PKC substrate motif antibody shows a significant reduction primarily in the staurosporin treatment. In panel B, we have another basophilic motif antibody, AKT. The AKT substrate antibody shows significant reduction in both MET inhibition and with staurosporin treatment. In panel C, we can also see significant reductions in phosphorylation in lane 3 with the AMPK and PKT substrate antibodies. Again, another 2 basophilic motif antibodies. And then in the last panel, in panel D, we can see that the MAP kinase motif antibody shows a modest increase in phosphorylation in lane 3 And I'll remind you, that's also we saw an increase in ERK phosphorylation in the multiplex western. So in total, these phospho-motif antibodies are not only useful for peptide capture, they can be also used to monitor the efficacy of treatment. In the next panel, we can see some of the qualitative mass spectrometry results in figure 6. And here we can see the phosphotyrosine, the basophilic, the serine-threonine mix, the metal affinity IMAC method, along with the proline mix. Each sample was run as an analytical replicate, and we've identified many thousands of phosphopeptides at a low false discovery rate. And I also mentioned that we also do not just qualitative identification, but quantitative information, and that's shown here on the next panel. On figure 7 is a description of how we do our quantitation based on the peak height or peak area. And this gives us a significant amount of information at the site level, which I'll show you in a few moments. But as you can see for the example here, this peptide is derived from MET, and it can show a significant reduction in phosphorylation using the MET inhibitor. Now, because we've identified and quantified so many peptides, we can get a global view of what's happening with phosphorylation in these cells. And in figure 8, we can see a number of scatter plots. The top scatter plot is for the cells that are treated with the MET inhibitor, SG11274. And you can see a significant number of peptides are showing decreases in phosphorylation, as you can see in red. And some are showing a modest increase, and that's in green. In contrast that to the fungal metabolite, or staurosporin, where there's a much more modest effect on phosphorylation. Again, with the basophilic enrichment, we can see significant reductions with staurosporin treatment, as we'd expect, and more modest changes with the MET inhibitor. And as we mentioned previously with the MAP kinase pathway, with the proline results, we saw an increase in phosphorylation that's very clear here in the staurosporin treatment, and and clearly less so in the SG11274. And then not only would the quantitative proteomics allow you to have a wide overview of what's happening cell-wide, we can also focus in on specific sites of phosphorylation, as we can see here in figure number 9. So here we can see what's happening at specific sites of phosphorylation on the receptor tyrosine kinase MET. Some sites are showing almost no change in phosphorylation, others significant 20-fold changes, while the center of the activation loop, those tyrosines undergo a 100-fold reduction of phosphorylation using the MET inhibitor. Now, putting all this data together, we can make some qualitative conclusions, and we can see that in the next figure, in figure 10. And here we have Venn diagrams that capture the qualitative information. So, for example, here on the left-hand side are all the antibodies used together, or the IMAC panel. And we can see there's very little overlap, suggesting that there's two classes of peptides are being enriched using the two different methodologies. It also suggests that these methods are very complementary. And on the Venn diagram on the right, you can see the phosphotyrosine antibody picks up the majority of the phosphotyrosine sites. The metal affinity will isolate some phosphotyrosine-containing peptides, but not the many thousands that we get with the antibody. We can also see that each antibody mix, whether it be the basophilic, the proline, or the serine-threonine mix, each of them isolates a separate group of peptides from the IMAC. And so the important point here is that there's a high degree of complementarity between the two methods. And looking more closely at the results from using the phosphospecific antibodies themselves, we can see here that there's a nice overlap between the serine-threonine mix and the proline mix of about 20%, and less overlap, as you might expect, between the basophilic and the proline, because they're isolating different types of phosphopeptides. Another advantage of doing proteomics, doing quantitative proteomics, is the isolation of changes in phosphorylation within a particular class of site. So, for example, here in figure 12, we can see that in the AKT family of peptides. We isolated peptides showing increases and decreases in phosphorylation depending upon whether they're treated with a MET inhibitor or with staurosporin. And finally, the results of this work can be plotted out in known canonical pathways. So for example, here in figure 13, we can see the phosphotyrosine cancer pathway populated in red by proteins that are shown to be changed in phosphorylation with our study. And likewise, we can see in panel B, the MAP kinase pathway is well populated using the proline mix of antibodies that I showed you here in figure 11. So in conclusion, the PTMScan method shows a high degree of complementarity using either metal affinity or the antibodies in combination. And the antibodies themselves are very useful reagents for pre-screening your sample to determine efficacy of treatment. And the method itself is very flexible. It can be used on plants, tumor tissue, cells. A wide variety of biological sources can be used as a substrate for the PTMScan method. Thank you.